Hello everyone, thank you for joining me today for this talk entitled An Innovative Product That Goes Beyond the Concepts of Pre, Pro and Postbiotics. As you may know, I'm Marco Ruggiero, I hold an MD and PhD, and this product I talk about is called 1VAR, the one and only panbiotic. And the talk and the product are made exclusively for Danabiologics and Mr. Choi-Yuk. Before we start, please allow me to introduce myself. I obtained my MD in 1980 in Florence, Italy. I have a PhD in molecular biology and a specialization in diagnostic radiology. As a young postdoc, I worked at Burroughs Wellcome in 1984 in North Carolina, USA, and later as visiting scientist at the National Cancer Institute of the NIH in Bethesda, Maryland, since 1987. Later, I was appointed professor of molecular biology at the University of Firenze, or Florence if you prefer, in Italy, in 1992, where I worked until 2014. I am the founder of Silver Spring, a Swiss company specialized in research and development of innovative approaches and products, including 1VAR, the product I'm talking about today. Over the course of my scientific career, I published more than 240 scientific articles, co-authored, among others, by scientists such as Eduardo Lapetina, Stu Aronson, Jackie Pearce, Peter Duesberg, Henry Bauer, the late Dr. Jeff Bradstreet, Dietrich Klinghardt. One of my articles was sponsored by Nobel laureate Sir John Vane. My current research interests are in immunotherapy, oncology, and neurosciences. Also, before we start, please remember that no information in this talk is presented as medical advice. Standard of care for every disease must be followed, as well as rules and regulations established by health authorities. I wish to disclose my conflict of interest that is quite evident here, because I have designed a number of protocols and approaches to different diseases, and I developed the product, 1VAR, and the approaches mentioned in this talk. I wish to thank Mr. Choi-Yuk of Danabiologics for his priceless support. This talk is for him, this product is for him and his company. And also I wish to thank all those who will watch this video for the attention that they will want to give to this presentation. So, what is 1VAR? What makes 1VAR? Essentially, there are 5 components. Because 1VAR is an original entire ecosystem with self-assembling supramolecular structures. The components are zinc, an essential mineral, a proprietary culture of prokaryotes and eukaryotes with their theater of activity, a long-chain sulfated polysaccharide, LCSP, of vegetal origin, endowed with GC-MAF-like activity. More on GC-MAF later. And then there are 2 botanical extracts. The first one is a botanical extract with known anti-inflammatory, immune-modulating, anti-cancer, antioxidant properties. Then there is a second botanical extract that contains natural free phenolic compounds, fibers, macro and micronutrients with known anti-inflammatory, immune-modulating, anti-cancer properties and known to rebalance sugar and lipid metabolism. Another important point is that 1VAR is designed according to a proprietary, highly innovative procedure that optimizes molecular interactions between its 4 or 5 components, so to increase their respective activities. In other words, 1VAR is not the result of the addition of the activities of its 5 components, but the result of the multiplication of the activities of its 5 components. In other words, if the biological activity of each individual component of 1VAR were 10, the overall activity of the self-assembled components would be 10 by 10 by 10 by 10, 100,000. rather than 10 plus 10 plus 10 plus 10 plus 10 that makes 50. In other words, its activity is 100,000 versus 50. This synergism is obtained at the molecular level through carefully calibrated interactions associated with the distribution of charges on the surface of molecules, leading to self-assembly of entirely original supra- and super-supramolecular structures. As I said, more on GC-MAF in the next slides. If you do not know what GC-MAF is, I recommend that you take a look at this article that was published as a special report in a famous British magazine called What Doctors Don't Tell You. And actually, this is the cover of the magazine. So it had the cover of the magazine, and it is called GC-MAF, the superhero. Well, if you want to go to PubMed, which is the official website of the US government, the National Library of Medicine, the National Center for Biotechnology Information, and you just look for GC-MAF, you will find, among other things, that is proposing cancer immunotherapy in viral conditions from COVID-19 to HIV, and also in neurodegenerative diseases. For example, this article deals with multiple sclerosis. And I have published a number of scientific articles on GC-MAF that, again, you can retrieve from PubMed. Just look for Ruggiero GC-MAF, and you find a number of articles where I describe a number of effects of this molecule, as well as I explain its mechanism of action. Let's move to the third component, a long-chain sulfate polysaccharide, LCSP, of vegetal origin endowed with GC-MAF-like activity. This is just one of the many repetitive units of this polysaccharide. I don't want to go into the biochemical details because I know that they could be very boring, but just let me tell you that even if you are not a trained biochemist, if you look at the LCSP and at this other molecule that is chondroitin sulfate, I think that you can recognize that these two molecules are very similar. As a matter of fact, LCSP shows a high density of negative charges that are represented by the sulfate group and also the carboxyl groups that are uniformly distributed on the molecular surface. Now, why do I focus on chondroitin sulfate? What has chondroitin sulfate to do with 1-var? Is there chondroitin sulfate in 1-var? No, the answer is no. There is LCSP. But, as I have shown before, LCSP is very similar structurally to chondroitin sulfate. And this is important because I demonstrated, together with my colleagues, Dr. Stefania Pacini, and a great German doctor, Dr. Heinz Reinwald, in 2016, we demonstrated that chondroitin sulfate is responsible for the biological effects of GC-MAF. You can retrieve this article from PubMed, the official website of the United States government, more precisely of the National Library of Medicine of the United States of America. How do we know that the LCSP has GC-MAF activity? Not simply because it looks like a chondroitin sulfate. There have been experiments on colon cancer cells, and as we can see, there is a dose-dependent inhibition of the alpha-nagalase from colon cancer cells by this LCSP. Please notice these are micrograms, so very tiny amounts. So it's very effective in its GC-MAF-like activity against nagalase. But LCSP does more than simply being or having GC-MAF-like activity, because it has a number of other activities that contribute to the health-supporting properties of 1-var with particular reference to immune modulation. It has been demonstrated that it enhances the phagocytic and secretory activity of macrophages, induces the production of nitric oxide, more about this later, and cytokines like TNF-alpha, IL-1, interleukin-1, and interleukin-6. And to this point, it is worth noticing that I demonstrated that nitric oxide is responsible for the cancer-killing effects of GCMAF, as it will be shown in the following slide, where we will see how activated macrophages surround and kill human breast cancer cells thanks to the production of nitric oxide. So let me guide you through these slides. These were taken under a microscope. So these are big human breast cancer cells. These are other big human breast cancer cells. These little dark blue dots are macrophages. So you see how many macrophages they surround the cancer cells. So let's go to a higher magnification. So this is one human breast cancer cells, and these are macrophages all around it and also on top of it. And let's magnify even more. Again, one big dying human breast cancer cell surrounded by these little dark blue dots that are macrophages. And this is probably the most impressive one. This is the highest magnification. Can you see here the macrophages is drilling a hole inside the cytoplasm of the cancer cell. And this is the nucleus of the cancer cell, and the cancer cell is dying of apoptosis. That is, its DNA, it's all fragmented. So you see its DNA has all these little dots. It means it is all cut in pieces. And here you can see other macrophages that are drilling holes in the cytoplasm. This is the big cancer cell. So they're killing the cancer cell. How do they kill the cancer cell? Thanks to the production of nitric oxide. Nitric oxide is like a bullet that the macrophages use to shoot and kill the human breast cancer cells. And this is a common mechanism. I was the first to demonstrate and publish this almost 10 years ago. Now, you may say, oh, well, fine, all these may be interesting or maybe not, but is it working? How can we assess the efficacy of one VAR on the immune system? How can we be certain that it is working in a repeatable, objective way? Well, as I have said before, and as it has been demonstrated since 1997, so many years ago, when macrophages are activated, they release nitric oxide, a molecule that is essential for the killing of cancer cells and cells infected by viruses. As this old article writes, sustained production of nitric oxide endows macrophages with cytostatic or cytotoxic activity against viruses, bacteria, fungi, protozoa, helminths, and tumor cells. So this is a fact. When macrophages are activated, they release nitric oxide. So this is how we can demonstrate that one VAR works, by looking at the nitric oxide that is released by the macrophages. And how can we do this? Well, let's remember nitric oxide does many things. One of these things is to induce vasodilation. So now the blood vessels under the influence of nitric oxide become larger, wider, and there is increased blood flow. As a matter of fact, nitric oxide is used because of its efficacy on vasodilation, and this has been known for centuries, I would say. So now we have to find macrophages in our bodies, and we have to assess whether they release nitric oxide or not. Where do we find macrophages in our bodies? Well, essentially everywhere, because they are circulating. When they circulate, they're called monocytes. When they are resident in tissues, they're called macrophages. So you find macrophages in all tissues, but where are they highly concentrated? In the adenoid, in the tonsils, in the thymus, if we still have thymus, in the bone marrow, in the lymph nodes, but also, and this is important, in the spleen. So in our spleen here, on the left, we have a high concentration of macrophages. I mean, this is a common knowledge. Everybody who has studied anatomy or histology knows that there is plenty of macrophages in the spleen. And we can easily use this, study the spleen using ultrasonography. This is a common ultrasound machine This is a picture from my archive. So this semi-lunar area here is the human spleen. This is diaphragm, the upper pole of the spleen, the lower pole of the spleen. So this thing here looks like a half moon, or like a sickle if you prefer. This is the spleen. And we can study the spleen with an application of the ultrasound machine that is called echo color Doppler, that studies the blood flow. So let me show you the next couple of slides. They show you how release of nitric oxide from activated macrophages can be studied in the spleen. So this is before assumption, and you see only a few blood vessels, not much blood circulation in a normal spleen. But after the assumption, look at this, look at all these colors. Here, both the cortical vessels and the hilar vessels of the spleen, they are vasodilated, a lot of blood flow. So this indicates that there has been a huge release of nitric oxide by the activated macrophages. This is another slide, enlargement. So again, now you can recognize that this thing here is the spleen, and these patches of color indicate the flow of blood. Red is when it moves in one direction, blue is when it moves in another direction. We use the acronym BART, blue away, red toward. So it's not venous and arterial. It depends on the direction of where the blood is going, looking at the probe. But don't be bored by these technical details for radiologists. Also, we can assess the efficacy of 1-VAR using the immune status questionnaire. This is a questionnaire that was developed about 4 years ago in 2019 by these researchers. You can go and take a look at the original article. And this questionnaire can be given to people who are taking 1-VAR, and you can see numerically the improvement of their immune status by looking at this. And we are collecting data in this regard.